All right, let's do question 4. Humans belong to one of 4 blood groups. These blood groups are A, B, and O. And here is a pie chart that shows the estimates of the percentages of people from Africa and the percentages of people from Europe in each blood group. All right, so here is the table. We need to complete the table to give the percentage of each blood group. So the first thing that I see here is that AB represents, 5% of people have the blood group AB. And that means that this section here represents 5%. All right, so let's try and figure out what B is. So B, we have 2 of these sections. So that's 2 times 5, that would be 10. Right, and there is 2 ways of doing this for blood group A. We can count the sections for blood group A, so that's 1, 2, 3, and it looks like it's slightly less than a half here. So that would be, let's say 3 times 5, that would be 15, and then it's not adding 2.5, so maybe like add 2, so that would be 17. So that's 17 there, and we just want to double check to make sure that everything adds up to 100. So let's add up these numbers together, 68 plus 17 plus 10 plus 5, and that gives you 100. All right, let's do the blood group for Europe. So here, let's start with A. Right, so with A we have 1, 2, 3, 4, 5, 6, 7, 8. So that would be 8 times 5. 8 times 5 gives you 40. Let me just remove this and this one as well. Okay. Now AB, because it's not, again, not exactly right in the middle, let's just do 100 minus these numbers. So 100 minus 45 minus 40 minus 12, and that would be 3. So 3% of people in Europe have the blood group AB. Okay, part 2. Some people need a blood transfusion. Explain why people with blood group A cannot receive blood transfusion from people with blood group B. Okay, so I'm going to draw a little diagram here. So imagine this is the red blood cell of a person with blood group B, and they would have these B antigens, right? And so what would happen is with people with blood group A, they see this and they produce antibodies. I'm just going to draw some antibodies here. So these would be the anti-B antibodies, and they would bind, they would recognize the antigens here, they would bind to it, and that would cause agglutination. That's when everything kind of gets clumped up together, which is quite dangerous if that were to happen inside of another human being. So let's write this down. So individuals with blood group B, have the B antigen on their red blood cells. Therefore, the person would produce anti-B antibodies, which will then cause agglutination. So agglutination is one mark. You have to talk about the antibodies that get produced, and you have to talk about how there are B antigens on the red blood cell. So those are the 3 marks. A male with the genotype IAIO and a female with the genotype IBIO have a child with a genotype IOIO. Draw a genetic diagram to show how the child inherits the genotype IOIO. So this is personally how I would do it. I would draw out a Punnett square. So let's say this is the male. This would be IA. This would be IO. This is the female. This would be IB. This would be IO. Right, and then, whoops, A, B, oh, let me just write that properly. IA, IB, IO, IO. All right. Okay, the next part. Determine the probability that a child from these parents will inherit the blood group AB. So if you can see here, this would be the offspring that would have the blood group AB, and that is 1 in 4. So you can say 1 in 4, you can say 25%, you can say a quarter, that's fine. All right, let's do part B. The heart pumps the body around the blood. The diagram shows a human heart. This heart has a hole in the septum. So the septum is the wall that separates the 2 sides of the heart. So it says this means that the blood from the left side and the left side of the heart mixes with the blood from the right side. Explain why the person with a hole in their heart breathes at a greater rate than a person with a healthy heart. Okay, so let me just do a little bit of labeling so we can sort of figure out what's in this diagram. So here we have the right ventricle. Here we have the left ventricle. This is the right atrium. This is the left atrium. So we know that in the right side you carry deoxygenated blood. And on the left-hand side, you would carry oxygenated blood. And if these blood, and with the hole in the septum, the 2 chambers, the blood in the 2 chambers can now mix. So the deoxygenated blood and the oxygenated blood can now mix with each other. And because of that, you would have some of the deoxygenated blood go here. And so that means that you would have less oxygen that's traveling around your body. And so how does that relate to them breathing at a greater rate? Well, if you have less oxygen being delivered to your body, then your body compensates to try and bring in more oxygen and try and breathe in more oxygen. Okay, so let's write this out. So where should we start? The deoxygenated blood will move from the left to the right to the right side of the heart. Right, and this would cause the deoxygenated and the blood to mix together. And because of that, less oxygen gets transported around the body. And so in order to compensate, So, person need to breathe in more, or to breathe more, so that more oxygen get delivered. Okay, pretty much it. Okay, question 5. There is a difference in the volume of urine produced on a hot day and a volume of urine produced on a cold day. Design an investigation to determine the difference in the volume of urine produced in a hot day and the volume of urine produced on a cold day. Include experimental details in your answers and write in full sentences. So whenever it comes to designing an investigation, we want to look at the independent variable, the dependent variable. We want to look at the control variable, although it doesn't really mention any control variables on the marking scheme. You want to make sure you repeat the experiment, because the more repeats that you do, the more reliable your results would be. And lastly, you want to compare the values. Okay, so on a hot day, collect urine from a group of people throughout the day and measure the volume. Okay, and then you would repeat this experiment for a cold day. Now, in the marking scheme, it doesn't say anything about doing repeats or reliability. It doesn't really ask for control variables either. But it's always safe to just write these points down, especially if you're given quite a bit of space as well. So anyways, repeat this experiment for a cold day. Then compare the two sets of data. All right, let's move on to this part. So I've included a little diagram over here in the bottom left. The diagram shows a cross-section of a blood vessel in the skin on a cold day. Explain the changes in this blood vessel on a hot day. So I'm just going to enlarge this diagram so you can see. So during a hot day, what happens is that we have the arterioles here, and these arterioles undergo vasodilation. So they widen, and because they widen, it increases the blood flow so that more heat can be lost via radiation. So those are the marks that you need to include. I'm going to make this one smaller, so we need to write 3 marks for this. So the arterioles undergo vasodilation, which would increase the blood flow to the surface of the skin. Skin, therefore, more heat is lost via radiation. right, the next part. Describe how ADH, so ADH stands for antidiuretic hormone, regulates the volume of water in the body on a hot day. So if you recall, ADH is a hormone that is secreted by the pituitary gland. Right, and let's look at a little diagram so we can see how that works. So if you can see here, right, ADH, when ADH is present, what happens is that more water gets absorbed from our collecting ducts, so that more water gets reabsorbed, so that you end up having urine with less water in them. Okay, so this is how we would write out the answer. Let me just make this smaller. Okay, so the hormone, sorry, I spelled hormone wrong. ADH is secreted by the pituitary gland. And causes, I should say, causes the collecting duct to become more permeable to water. Therefore, more water is reabsorbed into the blood. All right. So here are their marks. You need to mention ADH is secreted by the pituitary gland, and that causes the collecting ducts to become more permeable to water, so that therefore more water is reabsorbed into the blood. So that's the 1, 2, 3, 4, 5 marks here. All right, question 6. Describe how bacteria can be genetically modified to produce human insulin. So I've included a diagram here so we can have a better idea of what's happening. So we've got, so in order to genetically modify bacteria, we need to use a plasmid. So a plasmid is a little small section of, a little small loop of DNA that bacteria has, and it allows for bacteria to share genes with each other. And we can use this technology that they have to our advantage. So what we can do is we can have the insulin gene here, so we locate it and we cut the insulin gene and we insert it into the plasmid, then insert that plasmid into the bacteria, and then cause the bacteria to multiply and undergo cell division. Okay, so I am just going to make that one smaller. So here are the things that you need to say. Okay, first, the plasmid is removed from the bacteria. All right. Then the insulin gene is located and removed using an enzyme, and this enzyme is called restriction enzyme. Restriction Okay, now you would use the same restriction enzyme to cut the plasmid as well. So use the same restriction enzyme to cut the plasmid. Ooh, my handwriting's getting messier. The plasmid, cut the plasmid, and insert the insulin gene. I forgot to mention that we need to specify the human insulin gene. That's important there. To cut the plasmid, okay, so moving on to cut the plasmid and insert the insulin gene. Would use DNA ligase. So this is another enzyme that you need to know. So DNA ligase is responsible for joining these two pieces of DNA together. To join the insulin gene and plasmid, then we would insert the now recombinant plasmid into the bacteria. So insert the into the bacteria. All right, question 6 is done. So let's go over the points again. So first, number 1, you need to remove the plasmid from the bacteria. So that's 1 mark. Then you need to locate the human insulin gene and remove it using restriction enzymes. So that is 1, 2, 3, 4 marks so far. It says use the same restriction enzyme to cut the plasmid open. That's the 5th mark. And insert the insulin gene. That's a 6th mark there. The 7th mark would be using DNA ligase and then inserting the recombinant plasmid, that's the 8th mark, into the bacteria. That's the 9th mark. All right.